Okay, hello everybody. Welcome to this symposium of functional genomics. It's about when you have a nice genome and then you want to do analysis and going back, for example, to the organism and reveal the connection between the genomic information and in a, for example, phylogenetic context, what has changed during evolution. So we will start with the first presentation from Saoirse, who will talk about the sea urchins, and she is in the lab from Veronica Hinman. And welcome, Saoirse Foley.
Hi, everybody. My name is Saoirse, and I'm going to talk to you today about Echinobase, which is the online hub for echinoderm genomic data. So, just a quick bit of background as to what echinoderms actually are. They're ancient marine deuterostomes, and the group includes many creatures that you might be familiar with, such as sea urchins, sea cucumbers, and starfish. The group is about 540 million years old, and as you might expect for a group of that age, echinoderms are highly, highly diverse. In fact, if you look at this phylogeny on the right and take note of Strongylocentrotus purpuratus, the purple sea urchin on the top, and Anacia japonica, the feather star on the bottom, it has been speculated by some researchers that these two organisms are about as distantly related to each other as humans are to some insects. So this is a very diverse group indeed. They're of particular interest to researchers, though, because of their developmental pattern, which is considered to be quite simple and rapid. They develop quite rapidly. And their genes are also pretty easy to manipulate, and this makes them excellent model organisms for experiments in embryogenesis and skeletogenesis. And one of the really cool things about echinoderms is their ability to regenerate. So if you cut the arm off an echinoderm, please don't be cruel, don't cut any arms off echinoderms. But if you do, they can grow that arm right back. And so regeneration is rapidly becoming quite a hot topic in biology. And it wouldn't surprise me at all to see future researchers flocking to echinoderm systems to try and understand this process better. And so what is Echinobase then? It's a hub where we put in genomic information corresponding to echinoderms. We host it there, and we launched a new version of the website earlier this year at echinobase.org. The phylogeny on the right is now annotated with which species we are currently supporting. So the orange boxes around S. purpuratus, the purple sea urchin, and Acanthastra flancei, the crown of thorns starfish, mean that these are fully supported. If you want to use any of the functions of the resource, you can use them for these two species. The other two with the dashed orange lines, Pateria miniata and Anacia japonica, are partially supported, so you can't do everything with them just yet, but we are working towards integrating them fully. And some of the things you can do using Echinobase include just accessing that data. If you want to download the genome or the proteome of these species, we have links available on the site for you to do that. We also have a JBrowse tab integrated into the website where you can visualize those genomes. And then if you search for genes of interest, you can be given a lot of information about that gene. So in particular, since I started working at Echinobase, I focused on generating orthology information for the genes, and as well as that, developmental time courses, quantifying the expression of genes across development. And I'll be talking about those in particular, but just to show you what the gene pages look like, I searched for FOXA1 here. You could search in theory for any gene you want, but this is what will be returned. So in the top right, you can see the developmental time course with hours on the x-axis in S. purpuratus development. Y-axis then are the number of transcripts per million expressed. And then as you scan throughout the page, you can see the synonyms to genes, the interactants, implications for disease, predicted functions. And on the very, very bottom is orthology. So you can see there's a predicted human ortholog to FOXA1. And this is the data that I've been generating. In order to do that, though, we've been really focusing on this little creature here, the purple sea urchin, which is what we call our reference echinoderm. So the latest version of this genome was released in late 2018, early 2019, and it's a very good quality genome. There's almost 40,000 protein models, 870 scaffolds. And so because the other echinoderm genomes are not quite as good quality, we use S. preparatus as something of an anchor organism. And what I mean by that will become clear in the next slide. So in order to actually infer orthology, we use this guideline set up by the Alliance of Genome Resources. You can see on the top right that they have a stringent criterion, which we follow, meaning that they require at least 3 independent tools to converge upon an orthology before that call is made. And so you might even wonder why we care about orthologies. So they're predictably very useful in terms of experimental applications. So if you have a gene in a system that's difficult to work in, such as maybe humans, you can look up the echinoderm ortholog and then perhaps work with that gene in the echinoderm system instead. At Echinobase, we also use orthologies to name genes. So if we have an unidentified echinoderm gene that is orthologous to an identified known human gene, we can infer that they should probably be called something quite similar. The analyses we've actually performed so far include S. preparatus, the purple sea urchin, to human, and then S. perp to other echinoderms. So this is what I mean by calling S. preparatus an anchor species. So we don't do the other echinoderms to human. We infer the orthology between human and other echinoderms through S. preparatus. So there's a little bit of laterality there. As for how we actually detect these orthologs, I mentioned there are tools involved, and in fact there are many different kinds of tools that work in different ways. Some of them rely on reciprocal best blast hits, others rely on clustering, so they form little ortho groups of orthologous genes. And then there are phylogenetic methods that rely on external databases to create gene trees for any of the orthologous genes that we want to look at. And so to date at Echinobase, we've integrated a pipeline to return one-to-one orthologs from 5 different tools. The people at the Drosophila orthology prediction tool, they run a suite of 13 different tools. And we really actively want to try and emulate that system. So we are working to integrate more and more prediction tools. And as a consequence of that, we are only dealing in one-to-one orthologies right now, because many-to-one cases, of course, might change as more information comes in from more tool outputs. But here is the results of the orthology analyses so far. With just 5 different tools, we've recovered between 5,000 and 6,000 different orthologs between S. preparatus and 2 other echinoderms, including in the middle, you might note Anicia japonica, which is quite a very distant relative to S. preparatus. And then also over 3,500 orthologs between the purple sea urchin and human. And so, as I mentioned, we are trying to integrate more tools, and these one-to-one counts should indeed increase as more tools are integrated. And we're also aiming to include even more species in our orthology analyses. So we really want to focus on model organisms like zebrafish, C. elegans, maybe even your species of interest. We want to increase the laterality and to really enhance the usefulness of the database in doing so. I want to just end quickly by talking about how we use RNA-seq data at Echinobase to quantify gene expression across development. So this plot here on the right, it will show you FOXA in purple and AND1 in this brownish mustardy color. You can plot in whatever genes you want. We got this data from a 2014 paper. It released the RNA-seq data for each of these 10 different time points across the x-axis. And this tool, RSEM, can actually quantify the transcripts at each time point relative to the 5.0 version of the genome. These time points are also available as tracks on a kinase. And of course, this data is going to be useful for researchers who want to know when the expression of their genes is peaking. In summary, a kinase is expanding. Our orthology pipeline is good, but we're aiming to increase its number of tools and species. We use genomic data in interesting ways to host valuable information like the RNA-seq data for quantitative stuff. And we are really hoping to increase laterality with other organisms and databases. With that, I'd like to thank you for listening, and I am happy to take questions.
So thank you very much for this nice presentation, Saoirse. And it's indeed a very essential resource to have these. And optimal would be, of course, of many, many more clades we investigate. I think this is also the first time that I see a speaker changing the clothes directly after the lecture. Okay, I see this will happen more often here. Okay, we have several questions here, and one is from Bastian Fromm, and he is asking if you also annotate the non-coding genes in the genome.
Okay, sure. Thank you very much again for the opportunity to speak, by the way. But regarding this question, we annotate them insofar as we give them names. So at a kinase, we have a whole team dedicated to all of the different functions that you can see. So we will annotate them in terms of putative functions and names. But in terms of orthology, we're just using proteomic data. So if it's non-coding, it won't show up very readily there. So to some extent is the answer, yes.
Okay, we have another question, which is addressed from people outside the field of these intense functional genomics area. So is there a plan to also add phenotypic or geographical information associated with these different species into the database?
Oh, for sure. Right now, we are working on an ontology, a phenotypic ontology, phenotypic differences aspect for the database, where you can go in and look at whether or not some species will have a certain feature during a given point in their development. So yes, that is something that we are actively integrating as well. As for geographical differences, we have that information. So Strongylocentrotus preparatus is found all along the west coast of the US, Acanthaster plancy along the western Indian Ocean. So we have a good idea about where to find these species. And if there is interest, yeah, that's definitely something we could integrate too.
Okay, and I have a short question. So are you in touch with the sister group, like, for example, people who work on hemichordates who also get genome sequenced? And do you plan to do something common or add also hemichordates to the database?
As for hemichordates, I don't believe we're in contact with anybody at the moment, but of course, one of the key points from my talk, I think, is that we are really hoping to expand this resource as much as we can in the coming months and years. And so we want to increase laterality between all organismal databases and make this a resource, not just for echinoderm researchers, but for anybody who's studying something around development or some sort of cell biology question. So I think looking at hemichordate databases would probably be a good place to start in the coming weeks.
Okay, thank you very much. I think we move on now. Thank you for these questions and answers. And Andreas will now continue introducing the next speaker.
Okay, thanks, Andreas. Hi, I'm the other Andreas, and it's my pleasure to introduce Paolo Devana, a postdoctoral associate in the lab of Sonia Vernes at the Max Planck Institute for Psycholinguistics. And it's fantastic that we just had a question about the annotation of non-coding genes because he's going to focus on microRNAs across bats. Okay, Paulo.
Hi all, today I will show you how we used bat genomes to understand the evolution of microRNAs in bats. Bats show a range of exceptional adaptations like self-powered flight, echolocation, vocal learning, extreme longevity, and unique immunity. In order to understand the molecular basis of these traits, the Bat1K consortium sequenced the genomes of 6 bats that were released in July this year. These are highly contiguous genomes with excellent annotation that resulted in 99.8% BUSCO completeness, surpassed only by the human and mouse genomes. Having such complete and high-quality genomes also meant that we could annotate non-coding RNAs, including an important regulatory group, microRNAs. MicroRNAs play a role in gene regulation in virtually all organisms studied to date. They are short RNA molecules, in most cases between 18 and 22 nucleotide long, that mediate post-transcriptional repression largely via the interaction between the seed sequence and the target transcript. Repression mediated by microRNAs acts in all cells to determine their physiological properties and their identity, and it's crucial for cellular differentiation and stereotypical development. MicroRNAs can be conserved when they are found in all species, non-conserved, or species-specific. Our aim was to identify bat-specific molecular adaptations, and as such, we focus on bat-specific microRNAs and variant microRNAs, which are conserved microRNAs that carry nucleotide variants in the seed sequence in bats, an extremely rare event given its functional relevance. In fact, we identified 3 functional bat-specific microRNAs and only 1 microRNA with seed variants, MIR337-3P. This microRNA carries 2 changes within the seed sequence in all bats that are not found in any other mammal. Because the 2 variants occur within the seed sequence, we hypothesized that this should change the specificity of MIR337. To experimentally test this hypothesis, we devised a system that responds to the expression of a given microRNA in a specific and sensitive manner. This system makes use of a luciferase reporter that carries sequences responsive to the microRNA under investigation as 3' UTR. We call these reporters sensors, and this system can be used in any cell line. In this experiment, the control condition is a luciferase gene with no sensor. This condition represents the baseline as it cannot be repressed by any microRNA. Then we made a reporter with a sensor specific to the bat microRNA and one specific to the human microRNA. When overexpressing the bat MIR337, we observed a repression of around 40% for the reporter with the bat sensor, but no repression for the reporter with the human sensor. And vice versa, when we overexpressed the human microRNA, only the human sensor was repressed. This demonstrates that the 2 genomic changes affect target specificity. In fact, if we look at a pathway level, the change specificity results in regulation of different pathways by the human and bat microRNA, pointing to a new role for MIR337 in this clade. We now know that the 2 variants we identified in the bat genome affect the function of this microRNA. However, this can only have consequences for the organism if the microRNA is expressed. So we then looked at the expression of this microRNA across mammals. As you can see here, MIR337 is more highly expressed in bats than any other species by at least 1 order of magnitude, suggesting that this variant microRNA may have undergone positive selection. But while looking at the expression data, we saw something quite surprising. In most of the small RNA sequencing reads, we found a mismatch between the RNA and the genome. Where the genomic sequence carries an A, most of the reads had a G. At first, this was shocking because we didn't expect any mismatch. So we checked and it wasn't a problem in the genome sequence. It's something that happens at RNA level. So from shocking, this became exciting. This microRNA is edited. This results in two microRNAs potentially produced. One that I will call canonical miR37, which is the one I have characterized until now, and the edited isoform. Because editing also occurs within the seed sequence, I tested its functionality in the lab. New sensors were designed to enhance specificity against the canonical or the edited BAT miR37. And with the overexpression of the canonical miR37, we observed a repression of around 50% for the reporter with the canonical sensor, but no repression for the reporter with the edit sensor. And vice versa. With the edited microRNA, only edit sensor was repressed, resulting in completely different target specificity due to editing. It's like these are now two different microRNAs. And in fact, prediction of the genomic targets for the two isoforms of miR37 shows that the majority of loci targeted do not overlap. This dramatically changes the impact of this microRNA and expands the number of genes regulated. So how does editing occur? What is that gets edited? Enzymes called ADARs edit adenines that are found in double-stranded RNA in a bulge, and the sequence UAGA is the best substrate for editing. This is the human miR37 precursor. The hairpin, typical of precursor microRNAs, provides the double-stranded RNA, and there is a bulge with the sequence UA-UA. This A is edited in approximately 12% of the reads. The same bulge is present in BAT miR37, and this time the sequence reproduces exactly the most optimal substrate for ADARs. This is because of the genomic change between bats and other mammals indicated by the arrow. In fact, this results in 70% editing rate, which is a massive increment compared to all other mammals. I then went on to prove that this editing is actually mediated by ADARs. I used the same sensors as before, but this time I overexpressed only the canonical BAT miR37 with or without ADARs. As we have seen before, in the absence of ADAR enzymes, the canonical miR37 is unable to repress the edit sensor. So what happens with ADAR1? The expression of ADAR1 allows the canonical microRNA to repress the edit sensor, showing that editing has occurred. This means that normally, when the canonical miR37 is produced, only one set of loci is targeted, but the presence of ADAR1 results in editing that expands the range of targets thanks to the simultaneous presence of the two isoforms. So ADARs represent an extra layer of control for the activity of this microRNA, and this is a consequence of the genomic variant that arose in bats. To wrap up, I showed you that thanks to high-quality genomes, we identify a variant microRNA, miR37-3P in bats, and that this variant results in a new set of targets regulated in bats compared to humans. In addition, this genomic change has a knock-on effect, enabling ADARs to edit miR37, which in turn results in the expansion of targets regulated by this microRNA. By elucidating the molecular consequences of the variation of miR37 in bats, my work shows the importance of looking at the non-coding part of the genome to reveal functional variation that will lead us to understand the evolutionary roles of regulatory mechanisms. Of course, all this work wouldn't have been possible and so enjoyable, actually, without all the people in the Fairness Group at the Max Planck Institute in Nijmegen, the Netherlands, and all our collaborators. I also want to thank the organizers for inviting me and you all for the attention. I'm now happy to take your questions.
Okay, thank you, Paolo. Going through the questions, first of all, Sebastian Fromm asks, have you measured the absolute levels of microRNAs in different tissues?
Andreas, hi all. Yeah, thanks for this question. Actually, we did do some small RNA sequencing across several tissues in bats and also across the 6 bats that were already published. And what we find is that in bats in general, as you've seen already in the slide, MIR337 is very highly expressed. And this is compared to other mammals. But this is also, there is some differences across tissues within bats. And what we know at the moment is that MIR337 seems to be more highly expressed in brain areas compared to peripheral tissues. We have tested several brain regions, cortex, striatum, and cerebellum, and also liver and kidney. And in all cases, MIR337 is expressed, but more so in the brain.
Okay, thank you. And there are 2 related questions from Philip Schiffer and Peter Holland. So basically, can you tell if microRNA editing is more common in bats or conversely across bats than in other mammals, in that it would affect more microRNAs?
Right, so from preliminary data, I can tell that there is no significant difference between bats and the other species I've analyzed. So there doesn't seem to be really a difference in the machinery itself or at population level in terms of how many microRNAs are edited. But this effect seems to be boiling down really to the specific sequence that is the substrate for editing. And what we saw for MIR337 is that the genomic change is really creating this perfect site for ADARs, which then is increasing the efficiency of this enzymatic process.
Okay, thanks. And then finally, did you try the luciferase assays in bat cells, human cells, or both? And if so, do you think that would make a difference?
So yeah, this is actually a good question. We've been using this system for quite some time now, and we know it's really robust. And in the past, we've compared the effects for the same type of sensors across mouse and human cell lines, and we can tell that there is no difference depending on the cell line you're using. So one of the strengths of this system is that it can be, it's using the cell line mainly as a test tube. As long as the cell line expresses the machinery that will process the microRNAs, then there's no problem. Every cell line will work, and virtually every mammalian cell line is going to have this machinery. What can affect a little bit the final result is, of course, the background, whether that cell line expresses the endogenous microRNA itself. So if you're testing for, I'm saying MIR9, for example, that is a microRNA that is known to be highly expressed in the brain, a cell line that is derived from the brain will already have enough microRNA to reduce the luciferase, the reporter that you find, well, specific to MIR9. So the sensitivity will change a little bit, but specificity will not change.
Okay, thanks again, Paolo. All right, and we'll move on to the next talk.
Thank you very much.
Yeah, let's move on. This time we move over to mosquitoes, also flying objects like bats. And we also move on to Alice Jarvela. And she will talk about genome editing in mosquitoes and developmental gene-related genome editing. So welcome, Alice.
Thank you for the opportunity to talk about this work today, which takes advantage of the ability to use CRISPR and non-model insects to investigate a mechanism underlying gene loss. And the gene that I explored was this gene called paired, which we know is essential for segment patterning in a wide variety of insects. In Drosophila, where we know the most about segmentation, what happens first is early in the embryo, this gene engrailed is expressed in 14 stripes, and this prefigures the morphological segments that you'll see in the larvae later on. When paired is mutated, you lose alternate engrailed stripes, and correspondingly you lose alternate segments in the larvae. We see the same thing when we look at beetles. If you knock down paired by RNAi, you lose every other engrailed stripe, and the resulting larvae is also missing every other morphological segment compared to the wild type. It's very similar in mosquitoes. There are 14 stripes of engrailed in the early embryo, and a very typical larvae with 3 thoracic and 10 abdominal segments. But the crazy part is this is already the paired minus situation, because paired does not exist in the genome of mosquitoes. So in our own BLAST searches and phylogenetic analysis, looking for paired, we only were able to recover our reference genes from known species. But what we were able to find were orthologs of mosquito gooseberry, which is a closely related gene to paired. They're both in the PAX37 transcription factor family. And this is interesting because many years ago in the Knoll lab, they showed that gooseberry is able to perform the function of paired under the right conditions. So what they did is they had a paired mutant embryo that's missing every other engrailed stripe. And when they provided transgene where gooseberry is expressed under the control of paired's regulatory elements, they're able to recover these stripes again. Now, this was a highly engineered situation in transgenic flies, and we wanted to know whether or not the same sort of thing could also occur naturally over the course of evolution. So what we did was we looked at what gooseberry does in mosquitoes and if it has the same role that paired has in other insects. And so there are certain criteria that we wanted to see if they would be met. In Drosophila, we know that paired is expressed in a parallel pattern, which is 7 stripes, which I'll show you in a second. And we wanted to see if that same pattern appears for gooseberry in the mosquito. We know that in Drosophila, paired activates engrailed. So we wanted to see if gooseberry activates engrailed in the mosquito. And we know in Drosophila that paired's mutant phenotype is missing alternate segments. And so we wanted to see if we saw a similar mutant phenotype for gooseberry in mosquitoes. So first we looked at gene expression patterns. In Drosophila, we had already known from other work that paired and gooseberry have subfunctionalized by having distinct expression patterns. Paired is expressed earlier in stripes in the blastoderm. Gooseberry is expressed later in every segment. And what we see for the mosquito is a combination of both. We see this early striped expression, and we also see every segment expression later on. And that suggests that maybe this single gene is performing the function that's divided up amongst these 2 different genes in Drosophila. So to test these things, we made a CRISPR knockout mutant in mosquitoes. We replaced the first exon of gooseberry with a 3xp3 GFP cassette. What this did for us is it labeled the heterozygotes by eye color so that we could set up crosses. And so in our gooseberry cross, both parents are heterozygous mutants chosen by their eye color. And we also have control crosses where the male sibling is GFP minus. When we do this and we look at engrailed expression, what we see for the control cross is that all embryos have a 14 stripe pattern. In the gooseberry cross, where you would expect 25% of the embryos to be homozygous mutants, 25% of the embryos are missing every other engrailed stripe. Similarly, when you look at the larvae, our wild type larvae has 3 thoracic and 10 abdominal segments, which is a wild type pattern. But in our gooseberry cross, 25% of the larvae only have 2 thoracic and 5 abdominal segments, which is a pretty typical pair rule defect. So we see that all of the criteria for gooseberry replacing paired have been met. We see early pair rule expression. We see positive regulation of engrailed. And we see loss of alternate segments in our mutant condition. So what we think happened over the course of evolution is that paired and gooseberry subfunctionalize early on. So we know in many different insects, paired is expressed early and gooseberry is expressed late. And what we think happened in the mosquito is that gooseberry first acquired an early striped expression pattern. This was redundant with paired, and so then paired could be lost from the genome without any consequence for the embryo. I'd like to thank my lab, the PIC lab, especially Katie, who's an undergraduate who worked on this project, the Insect Transferation Facility for their assistance with the CRISPR injections, and the NIH for funding.
Okay, so thank you very much, Alice, for this talk. And we can now, the audience can now ask questions. And it's of course not a question that bats eat mosquitoes, but somebody has a question maybe? So there is one question from an unknown person. Have you looked at cis-regulatory differences in known or predicted binding sites for gooseberry or associated proteins?
That is something that I'm doing right now. I am trying to get injections done when I'm only allowed in the lab a couple hours a week, and we are collaborating with the insect transformation facility at Shady Grove. They're doing some of these injections for us, but long story short, we've done a fair-seq, which is sort of like a tax-seq, and we have some candidates that we're testing, some regulatory regions.
Okay, thank you. And one last quick question. So how much DNA are you able to isolate from one embryo?
DNA from one embryo? I don't know. I usually get, a few dozen in there and mix them up. I've never tried to just isolate DNA from one.
Yeah. Okay, good. So the likely person who works on small, tiny animals or even embryos. Okay, thank you very much. I think we have to move on to the next speaker, and Andreas will take over.
Okay, thanks, Andreas. So at this point, we are moving even smaller, and it's my pleasure to introduce Sarah Calhoun, who's a postdoc in the Grigoriev group at the US Department of Energy Joint Genome Institute, and that's at Lawrence Berkeley National Laboratory.
Hi, I'm Sarah Calhoun. I'm a postdoc in the Fungal and Algal Genomics program at the Joint Genome Institute. Today, I'm going to talk about a multi-omics pipeline that we've developed to profile novel algal strains for bioenergy applications. In our group, there is a growing effort to sequence new species of algae. We now have over 100 algal genomes hosted on our web resource, FICACOSM. JGI has sequenced about half of these ourselves, and the number continues to grow. We're interested in studying algae at the JGI because of their significance in the environment and energy applications. Through our community science program, we work with academic researchers and industry or national lab partners on projects that relate to all of these topics shown here. But the main focus of today's talk is on algae as potential biofuel feedstocks. Some algae produce large levels of lipids that can be used for biofuel production. In particular, the triacylglycerols, or TAGs, can be used as fuel precursors. Traditionally, development of biofuel production has been focused on a limited number of species. We're contributing to efforts for exploring the potential of new species and improving these species by collecting functional genomics information on them. This helps guide strain engineering to improve overall biomass production and general robustness when they're ultimately deployed in outdoor farms like the one shown here. This project is a close partnership between Berkeley Lab, where our team is led by Igor Grigoriev, and Los Alamos National Lab with Sean Starkenberg. Our goal was to develop a functional genomics pipeline as a platform for the production and interpretation of multi-omics measurements with the aim of accelerating strain improvement. Our initial target was a strain of Senodesmus from the National Renewable Energy Lab. This is a promising strain based off of its ability to accumulate biomass, and it produces a lot of lipids. So we've sequenced the genome and collected transcriptomics and metabolomics data, and by integrating this data together, we aim to identify regulators of carbon productivity to inform strain improvement. Together, this encompasses our functional genomics pipeline. We perform temperature perturbations as stress can increase tag levels. The transcriptomics and metabolomics measurements were taken in cold stress and heat stress conditions over the course of one day. When we look at the transcriptomics data, we see that the gene expression is more significantly altered under cold temperature. We performed large-scale analyses, such as building co-expression networks to look at correlated genes and identify potential regulators. We've looked at the changes in metabolite levels. For example, we see changes in the composition of lipids in the stress conditions. By applying this pipeline to our algal strain of Senodesmus, we have been able to observe changes that are consistent with the cold adaptation response in other algae and plants. For example, we see that lipids with more double bonds or a lower degree of saturation are increased in the cold stress. Then when we look at the gene expression data, we see there's an increase in expression of fatty acid desaturase genes in the cold samples. The fatty acid desaturases are involved in the desaturation of acyl chains and lipids. These can be relevant for biofuels because we know that lipid composition can have an effect on biofuel properties. Finally, we use our gene co-expression networks to identify transcription factors that could be involved in modulating these properties. So to wrap up, we developed and applied a functional genomics pipeline to profile algae. We've demonstrated this on a new strain of Senodesmus and identified targets for strain engineering that are being tested. Next, we are adding new types of data, such as proteomics, and we now have 3 new strains in our pipeline. With this pipeline, we are accelerating the process to characterize new species with high potential in biotech applications. Thank you for your attention, and I'd like to acknowledge all of my collaborators on this project. And lastly, I encourage you to check out the resources on Phycocosm for access to algal genomes and our web-based tools. Thank you.
All right, thank you, Sarah, for that great talk. So while the questions are still coming in on the chat, I actually had one myself. So I'm wondering if you've ever looked at variation within these populations. So obviously you're probably getting an aggregate over large numbers of algal organisms and cells. Do you think that some of the networks that you're finding could be driven by variation that exists within that population?
Yes, I totally agree. That's such an interesting question to start looking at the population of these algal strains. We actually haven't been looking at that so far, but our group has been developing a lot of methods to look more at pangenomes. And I think that in the future, that's going to be the next step, is to look at more, rather than just single genomes, pangenomes.
Okay, so let me ask another a follow-up question. So when you have these multi-omic measurements and you have these networks, and ultimately you mentioned that your goal is to do some engineering using CRISPR and other techniques. Do you have a sense of which nodes in these networks might be the most causal and might be the best ones to go after when you're deciding what exactly to edit?
Yeah, I think it's still development necessary to identify and predict with accuracy what are the most causal nodes. A lot of these network analyses, you can identify these hub genes. So these are the ones that have the most interactors associated with them. And you can begin by targeting those hub genes first as far as gene editing. So I think that's the approach we're going to be taking.
Okay, great. Yeah, that makes sense. Okay, and then Philip Schiffer is asking, in your co-expression networks, do you also look at connective nodes and interaction partners, that is genes that connect between others but that aren't upregulated?
Yeah, that's a good question. So we definitely are looking at a variety of different interactions. Sometimes you can get correlations that don't necessarily show like a significant differential expression using these standard like DE-seq tools. And so by moving to these co-expression networks, you can see correlated genes. And by using orthogonal methods, we can kind of narrow down further these gene interactions.
Great. It's interesting that you're able to capture those relationships. In the meantime, there are a number of other questions coming in. So if you have the time, it would be great if you could answer some of them within chat, especially since I used up so much of your question answering time
no
myself.
worries.
But in the meantime, I'll pass it back to the other Andreas, who will introduce our last speaker.
Thank you.
Okay, yeah, we are coming to our last speaker, and people might think we are a bit biased about flying objects, but these animals are flying again. So it's about stalk-eyed fly, very fascinating animals, and genes, differential expression of genes related to aggression. So Sebastian Pohl will talk about it, and he is from Yale NUS College.
Hello, today I will talk about aggression and differential gene expression in a Bornean stalk-eyed fly, Teleopsis polyphasis. Aggressive behavior is a basic behavior in most animal species, and it's usually displayed to acquire or defend resources, such as food or access to mating partners. A famous example is the fighting behavior in male deer, where they try to compete for females. In insects, the genetic basis of aggression has been studied extensively in Drosophila, and this has usually taken place in highly controlled lab experiments with mutant strains of flies. Here we have performed a field study on aggressive behavior in stalk-eyed flies, followed by RNA sequencing to detect differentially expressed genes. Our study species is Teleopsis polyphasis. This is a sexually dimorphic species. We have here on the left the male with the eyes all the way at the end of these long eye stalks, and the female on the right also has eye stalks, but they are not as prominent. The males secure access to specific sites, such as the surface of broad leaves, and when they encounter a different male, they will line up and then either fight or fly away if they are smaller than the other one. The winners then gain mating opportunities with visiting females. Our field site is in Borneo, in the primary rainforest at the Kuala Belladong Field Studies Center in Brunei, shown here on the left. And on the right, you can see a typical habitat where we find these flies at the side of the small creeks. We catch these flies in the field, and then we will measure them. This is like a typical size. And then we record fights with size-matched triplets in experimental arenas. So the drawing here on the left shows this experimental arena with two flies inside. And on the right is a fly in a control vial. This is an example of such a fight. On the left, we have a fly on a piece of banana peel. And on the right, we have another fly approaching. And you will see now here some of the fighting behavior. So we see some tussling with the arms. We see some abdomen bobbing. And then eventually, we will also have some direct attack. So after we recorded these fights and determined the winner and the loser, we freeze the flies and we preserve them in RNA later for RNA sequencing back at the lab. We perform trisel extractions of RNA from heads and thoraces, and then followed that up with Illumina sequencing of the heads and the thoraces of over 48 individuals. And we added additional testis sequences that were obtained earlier to build an assembly as complete as possible. To build the assembly, we used the Trinity pipeline for de novo assembly with Callisto. And we blasted our results then against the Teleopsis dalmani reference cDNA assembly. After quality control and filtering, we retained 9,800 transcripts of originally almost 70,000 successfully blasted transcripts. And we then performed the seq for 36 head sequences from 12 triplets. Our cutoff criteria were an FDR of 0.05 and an absolute log 2-fold change value of bigger than 1. Here on the right, we see the heat map of the results, and we have the samples in the columns and the genes in the rows, and we find 18 differentially expressed genes. 15 genes were upregulated in winners relative to the controls, and 13 of those genes were also upregulated in winners relative to losers. 2 additional genes were upregulated in winners compared to losers, and 1 gene was upregulated in losers compared to winners. When we look now at the individual level, we see that the winners, shown here in red, cluster mostly with winners, whereas the losers, shown here in green, cluster mostly with the controls. This clustering is not perfect, but we can see that pattern. When we look at the DE genes of interest, we see that among those upregulated in winners were catalase and aldehyde dehydrogenase. And these are both part of the tryptophan and serotonin pathway. And it has been demonstrated before that high serotonin levels correlate with higher aggression levels in Telopsis dalmani. So to conclude, we can say that winning makes a difference in these flies when it comes to gene expression. And that studying transcriptomics in situ, so in a field setting with wild-caught flies, still allows us to find differential gene expression patterns despite a lack of standardization. And with that, I'm happy to take any questions.
So thank you, Sebastian, for this nice presentation. And I found the video of the fighting flies very cool, as several other people in the audience. So we can now ask you questions. So at the moment, I don't see any, but I can take a question from which was asked to the previous speaker, Sarah. So are there any economically related genes expressed differentially?
In the flies, I don't think so. I mean, we are still trying to figure out more details of which genes are differentially expressed between the fighters and looking at the different effects. I don't think it's anything that's commercially interesting, but it's really interesting to be able to match the behavior with the differential gene expression.
Sure. So now questions appear. So what were the completeness BUSCO scores and what database was used?
So we used, so for the annotations, we used an ensemble database. So there's a reference genome on Telopsis dalmani, which is a closely related species. And that is where we blasted our results against.
Okay, so I will ask you a question. So what kind of aggression you plan to relate this behavior to? So is it only insects or is it other animals?
For now, we're looking at the flies, and then it would be interesting to see if we find some more general patterns. So if we can find some genes that are involved not only in insects, but also in other animals. I mean, we've heard some of the talks in the session that there might be some genes that are very well conserved and that we can track over like a long evolutionary history. So it might be possible to find some genes there.
Yeah. So we have one question from Kristen Panfilio. Is there a genomic basis for predicting which flies may upregulate the serotonin pathway more?
Oh, that's a good question. I don't know if there's anything known about that specifically. What we're trying to show here mostly is that we can find these differences like in a field setting, basically. So we don't know much about the genetic background of the flies. I mean, they can be old flies, young flies. We don't exactly, we don't know what they have been doing before. That is the situation that we find actually in nature, because if you're a fly and you find someone else that you have to fight against, you also don't know what is their history. Yeah? And so that's basically the intention, to see what kind of differential gene expression and what kind of differences we can find if we are in a more natural and not so regulated setting.
I'm waiting for further questions to appear. Yeah. So if you manipulate the serotonin levels, can you affect aggression? And how do you manipulate the serotonin levels?
Yes, there are actually studies that have been done in different species, and we have been performing those. We haven't analyzed them yet, so we have done some preliminary experiments in our species. You can add some precursor, so 5-HT is a precursor of serotonin, and so you can apply that basically in a solution. You put a little drop on the abdomen of the fly, and then that gets introduced into the body, and you can then actually show that it changes the serotonin levels in the flies and changes their behavior as well. So that's why we were quite happy to see like genes that are connected to serotonin even in these non-manipulated flies.
Very interesting. So there are no more questions appearing at the moment. Always when I say this sentence, suddenly there are several popping up. This is not the case here now. And so I will hand over, so for the closure of the session, to Andreas, the other Andreas, as everybody knows by now. And so he will close up, and thank you everybody for the speakers for your presentations. I enjoyed it a lot. So Andreas, please.
Okay, thanks, Andreas. I'd also like to thank all of the speakers for a wonderful set of talks. So we purposefully really wanted to get talks from a wide variety of different organisms and try and make some connections across those organisms, ranging all the way from mammals in terms of the bats to algae and everything in between, so to speak. There's also really a diversity of different techniques and technologies used, focuses on coding, non-coding, and regulatory sequences. But I think there are also, among these differences, there were a lot of commonalities. So one of the things that I took away from it was the importance of annotations as a foundation for the biology, and also the importance of genomic tools and resources for answering some of these questions. And I also think it's quite interesting that even that across multiple of these organisms and talks, there's also been a focus on genome editing and other applications to go from some of the associations that exist between some of the annotated elements and behaviors or phenotypes, to go from these associations to actually being able to interrogate causal mechanisms and understand some of the biological bases of these traits. So again, I'd like to thank all of the speakers and thank all of the participants for asking some fantastic questions and joining us here. And I think next on the agenda is everybody's getting back together for a concert that should be in the plenary stream. So thanks everybody, and enjoy the rest of the conference.